presentation is entitled A Potential New Source of Rust, Puccinia arachidis, Resistance from Wild Peanut Relatives Arachis stenosperma, and Arachis batizocoi. I'll have you say that more better than I did. So, and by presenter Kate Costello. So thank you for being here.
Thank you, Dr. Kemerate. Yes, it's batizocoi. It's kind of a hard one to say. So yes, I'm Kate Costello. I'll be presenting my project on rust today. Let's see. So to start, I just want to talk about the origin of cultivated peanut. About 9,400 years ago, wild peanut, Arachis duranensis and Arachis ipiensis bred together to create a hybrid, and then had spontaneous chromosome doubling to create the AABB genome that we know today. And then along with that, there was over many years domestication and selection to finally get to our well-known Arachis hypogaea peanut that you can see on the right, our nice big seeds that everyone likes to eat. So wild peanut relatives such as batizocoi and stenosperma, which is what I'll be talking today, have been found to have a lot of really good resistances to lots of different pathogens and pests. But today specifically, I'm going to talk about fungal diseases, which is peanut rust. So in order to be able to breed wild and cultivated peanut together, there's a couple steps that have to happen. After the chromosome doubling in 9,400 years ago, the cultivated peanut became a tetraploid, whereas a lot of wild peanuts are diploid, meaning that they are not sexually compatible with each other. So luckily there's a way for us researchers and breeders to be able to breed these together. So in this case, we used batizocoi, which is the KK genome, and stenosperma, which is the AA genome, and bred that together to create a F1 generation. And then you can use colchicine on these little seedlings to create a spontaneous, synthetic chromosome doubling, which will then allow us to have a plant that has the tetraploid genome that can then be bred with cultivated peanut. So in this case, we ended up with a plant that we call batsten1 for batizocoi and stenosperma, and it is an AAKK genome that can then be bred with the cultivated peanut, which is AABB. So for my project, I was working with a really great BC3 population, backcrossed population that was created for root knot nematode resistance. As you can see in these pictures, there's a lot of susceptible cultivated peanuts out there that are really susceptible to nematodes, and they will die in the field if there isn't treatment for that. Versus some of these resistant cultivars that have resistance from wild peanuts. Like I said, the wild peanuts have a lot of resistance in them that cultivated peanuts don't have. So here's an example of one that has resistance from Arachis cardinacei. For this project, someone from our lab who graduated recently, Dr. Carolina Ballantaborda, she used a marker-assisted backcrossing scheme using that batsten1 peanut I was talking about before to create a population that was resistant to root knot nematode, specifically with these chromosomes A2, A4, and A9. Now, where I come in is that I wanted to know, are these plants that are already resistant to root knot nematode and that have various introgressions from the wild peanut in its DNA, are they resistant to leaf spot and fungal diseases as well? So what I did was I chose some families from within her population. So basically I chose some seeds specifically from her population that had been genotyped. So I could look at how much DNA from the wild batsten1 was still within the genome of these families. And so I could decide which ones I wanted to use to look at for rust resistance. So I decided to choose some that had a very low amount of wild DNA, such as this, what I call seed 8 and then seed 7, and one that had a very relatively high amount of wild DNA. At this point, BC3 does not have a very large amount of the wild DNA in there. So 15% is considered a high amount in this case. Basically just so that if I found resistance in these low families, I could say, okay, there's only a few segments that are integressed in this genome from the wild peanut. So it's way more likely that this is what's causing the resistance. Versus seed 11, which had a much higher amount of DNA, it would be a lot harder to tell what was actually causing the resistance. But then they could also be a comparison to each other. So just a little bit of background on rust, Puccinia arachidis. It causes these orange pustules where the spores erect through the lower leaf surface, as you can see in these pictures. It doesn't cause defoliation, but the leaves do dry up and it definitely reduces yield. It's a problem around the world, especially in warm, humid climates, which means mostly countries in the Southern Hemisphere, but then we've also got some of the Southern states, including Georgia here in the US. So this disease can cause up to a 57% yield loss. To look at my plants and find out if they were resistant to the rust or not, I did a detached leaf bioassay. This is a much more controlled way of doing it than trying to do it in the field. I collect leaves from each of the plants that I want to test, create a spore suspension from rust. It's just water and rust and a little bit of tween. And then I paint that spore suspension onto the underside of the leaves because the underside is where rust is mainly going to be infected on the leaves. And they're in these individual little Petri dishes so that they have their own little moist chamber, and I can keep them at a high humidity and check them every day to see if there's any signs of rust on each of these leaves. And after about 3 weeks, that's when I usually started to see if there was going to be any rust or not on these leaves. So in order to rate the rust pustules that I saw on these leaves, I looked at them under the microscope after I finished the bioassay, and I used this index that my PI, Dr. Soraya Bertioli, created, basically looking, is it a very small non-sporulated pustule up to these very large super sporulated pustules that basically is a lot of pustules just so clustered together that it's hard to tell where one begins and the other one ends. And then we have this sort of rating that we call the index, which incorporates this rust pustule scale in order to say, okay, how much disease severity is there on each leaf? How many lesions are there? How many of them are actually sporulating versus the very small non-sporulating ones? So how many of them are actually producing secondary inoculum? So we call this the index. Here is some graphs from my assays on the, you can see this is seed 11, and it was very susceptible to rust. Every single plant and pretty much every single leaf from each plant just covered in rust. So this was a bit too susceptible. I mean, if I'm looking for resistance, having a family of plants that are all susceptible is not going to be very useful to me. So I also looked at this seed 7. On the other hand, seed 7 was too resistant. As you can see, I only had one plant from this family that had any resistance. This would make it really hard to actually compare which ones are resistant to susceptible, looking at their genotype and saying, what segments is causing this resistance? So luckily, seed 8 was perfect. The family segregated for resistance. Some plants that were susceptible had every single rep, every single leaf were covered in spores, whereas the resistant plants had just basically complete, every single leaf was resistant, so no spores on there. This will make it a lot easier for me to identify the segment within the genome that is actually causing the resistance. So here's an example from one of my bioassays for seed 8. As you can see in red, those are some of the plants that were susceptible versus all of these resistant ones that just had no spores on them at all. It just makes it a lot easier to tell that which ones are susceptible versus which ones are resistant. And I can look specifically within the genome and figure out what's going on. So from these results, we did Affymetrix genotyping to look at the genome of these plants that I used and look at them to compare which ones were resistant and which ones were susceptible, and decide which wild segments might be causing this resistance. So I have some preliminary results that seem to show a correlation between the wild introgressions of DNA on chromosome B2 and resistance to rust. So that's pretty exciting. Definitely looking forward to finding out more about what is causing resistance in these plants. So that I want to thank everybody in my lab and my committee. And if anyone has any questions, then I'd appreciate it.
Thank you very much, Kate. And we have time for questions. Any questions from the audience? I have a question because you're working with rust diseases. How has the problems with the COVID virus affected your research and your work outside of obviously what would be happening in Athens? Is with the disease, which is more of a problem outside of the state of Georgia than within the state of Georgia, working with a peanut disease like that, how has that affected your ability to do your research over the past year and a half?
Yeah, well, it just basically meant that we had to make sure to keep inoculum going within the lab, so that, because like you said, there wouldn't be as many chances maybe to get new fresh inoculum from other fields or, other people. So we were, keeping our cultures going in the lab.
It has been difficult for everybody.
Yeah, definitely.
Here's a question from Sham Tullery. The resistance on B2 was an introgression from K or the A genome?
That's a good question. And I don't remember off the top of my head, I'm sorry. But yeah, that's definitely a important thing to know. I need to, thank you.
And I believe that's all the questions we have now. Thank you so much, Kate, for participating in today's session.
We have one more.
Oh.
Another question, or?
We do have one more question. Came in at the wire. This is from Abraham Fulmer. Is there any chance that the inoculation method is there any chance that the inoculation method would lead to additional variability with your results? If so, what would you change, or how would you change it?
Right. So I guess you're wondering if perhaps some leaves get painted with more spores versus other spores or fewer spores. One thing we definitely do is that when I'm creating this suspension is I use a hemocytometer to, concentrate, make sure I have a very small amount of spores, basically the smallest amount that we have figured out in the lab that will create inoculation without overloading the leaves. And then as I'm painting them on there, pretty much just trying to, stir it around as much as possible and paint it on there. Yeah, I think it would be really hard to fix that a lot more to make sure that every single leaf gets the same amount. Perhaps trying to put exactly the same amount using a pipette on each leaf, but some of these bioassays are really huge and they take a long time. So adding some of those variables in would just increase the time even more. So I don't know, it's kind of a trade-off, I guess.
Okay, Kate, thank you very much. That's the time we have.